Welcome back, everybody. I hope you're able to grab a little snack and get ready for our next session of speakers. This next session will be 10-minute science talks from 6 of our AWG speakers. And the first will be Alex Co. from the University of Southern California at the Information Sciences Institute. Alex, if you are on, please go ahead and turn on your audio, video, and go ahead and share.
Cool. Can you hear me?
Yes, we can.
Awesome. Okay. So in this lightning talk, I want to share our recent effort to build Glare, open source representation learning framework to discover hidden patterns in spaceflight transcriptome. Before getting into details, I want to give a refresher to everyone. What is representation learning and why do we want to use it? When I explain about representation learning, I like to use this analogy one of my colleagues made, which is Plato's allegory of the cave. So in the cave, there's a bound observer chained in the cave, and they can't see what's behind them. So only way that they can see what's behind is through the shadow they could get from the fire. The observer will look at the shadow and think the object that is behind them is a vase, because the shadow looks like a vase. And the shadow would be the representation of the vase here. And going back to the space biology field, the truth object, which is a vase, would be spaceflight transcriptome. And the shadow, which is an observation, would be PCA, t-SNE, or other dimension reduction method that is widely used in the field. But we need better data representation for further analysis. So better representation learning means light bulb instead of fire, because it will provide clearer shadow of the objects. And we also want more detailed understanding of the representation by maybe giving the observer some different tools like telescope or microscope to look at the representation in different perspectives. So the current practice in the field, we use a lot of PCA, t-SNE, k-means to analyze the GenLab data set. And the main alternative would be autoencoders, which is the neural network-based model that could produce data representation that captures a nonlinear relationship within the dataset. And with that said, we introduce Glare, GenLab representation learning pipeline. So we start with GenLab process dataset. In our case, it is GLDS 120, namely a CARA dataset. I would love to explain more about this dataset, but for the sake of time, I will skip that part. But so we start with the verification study using data discretization and supervised learning methods to see if there's indeed a pattern within the data set. So we want to study more about the data set. And then our pipeline starts with pre-processing using clustering. It was the case for our CAR data set, but this could be different for different GenLab data set. And we perform representation learning. So our main contribution is coming from building sparse autoencoder, which I will explain with further details in later slides. But on top of sparse autoencoder, we provide options of dimension reduction methods such as PCA, t-SNE, UMAP for further investigation. And using this data representation, we perform ensemble clustering. So as I said, we want to give more tools to the researchers. So we perform 3 different clustering to achieve the consensus cluster. And then lastly, we perform post-pipeline analysis, including gene ontology analysis and SHAP analysis for feature importance and to induce interpretability. So verification study starts with data discretization, which is just reconstructing our data set. So this is our CAR data set. It was experimented on flight and ground control with three genotypes, namely Columbia, FID, and WS for Arabidopsis, and two light settings. So we reconstruct this unlabeled data set to have a label for the location. And we perform classification on different data models as we can reconstruct our data set to have location variables or genotype variables or light setting variables. And for the car data set, we indeed see the pattern with 91% of test accuracy. And we, as we verified there's some patterns in this data set, we move forward. Next we perform pre-processing using clustering. We start our initial investigation of the data set using classical or commonly used methods such as PCA and K-means. And for the car data set, 98% of the data we condense in this area. So we use this opportunity to detect the outlier and clean the data set a little bit more to produce better data representation. So data representation. So we compare, you can see the comparison of the different data representation of the data set for the flight data and ground control data from PCA, TSNE, UMAP, sparse autoencoder, and fine-tuned sparse autoencoder. So our sparse autoencoder consists of 3 blocks of encoders and 3 blocks of decoders. And we can get the data representation from the bottleneck layer. And the one, our building block consists of linear layer normalization and activation function, and the sparsity was induced with L1 regularization. So you could either train this model using your target data set, which was normalized counts of car data set for us, or you can pre-train this model using the single cell data set and fine-tune your target data set to add more context to your data representation. And as you see on the evaluation of the data representation, the fine-tuned sparse autoencoder has overall better quality than other methods. So ensemble clustering. So ensemble clustering, we use 3 different clustering algorithms, namely Gaussian mixture model, hierarchical density-based clustering, and spectral clustering, which is all based on different statistical methodology, which can provide different perspective. As you can see, the clustering results looks different for all different space clustering, but we perform ensemble clustering through evidence accumulation clustering, which is building association metrics through consensus voting and performing final hierarchical clustering to drive the final consensus clustering. And using this clustering results, we find the hypoxia cluster that was found only on flight data representation for the our data set. Lastly, we perform post-pipeline analysis. We take this hypoxia cluster to do some more analysis, such as producing heat map using normalized counts, or producing gene ontology analysis using Metascape. We also construct a stress knowledge map on 5 transcription factors that was found in hypoxia cluster to give the better insight that we found on the flight data representation. Lastly, we perform SHAP analysis, which is, so far we were more focused on comparing flight and ground control. But as I said, in car data set, there is 3 different genotypes and 2 light setting. And through performing SHAP analysis that derived from the model that we used for verification study, we can see how different experiments had different contribution on classifying gene as flight or ground control. You can also produce a summary SHAP plot to compare the overall contribution. So with that said, so you can use, this is SCLARE, GenLab representation learning pipeline. So you can use your GenLab data set to perform representation learning on your research. It is designed to easily integrate with your own data of interest within GenLab. And there's, or you can use for education resource, such as like GenLab for high school. And our source code are shared at OSCR GitHub here. and the preprints will be ready soon as well. And thank you for listening. And that's my presentation. Thank you.
Thank you so much, Alex. Just want to remind the audience that um, we're going to have Q&A at the end of the session. So once all the speakers have finished talking, all the speakers will come online where we can do a 10, 15-minute Q&A session. So when you add your questions to Conference.io, please add the name of the speaker so I know to whom to direct the question to. So with that, our next speaker is Maywish Riemann from the National University of Science and Technology. Unfortunately, she was not able to be with us in person today, but we do have a recorded presentation that we will play for her. So give me just a moment to load that up.
Hello, this is Navish Rahman, and today I will be presenting to you the differential expression of microbiologically influenced corrosion-related genes in Bacillus subtilis in low Earth orbit. So this was my project, and today I will brief you about it. So I am Navish Rahman. I am a master's student at Nust University, Islamabad, Pakistan, and this project was conducted under supervision of Dr. Faisal Khan. These are some of my handles. Twitter handle. If you want to contact me for more information, this is my Instagram handle and this is my LinkedIn handle. So now moving on to the next slide, let's give you an introduction about the project. So microorganisms exist in two modes. One is platonic mode and the other is biofilm. So under harsh or in stressful conditions, bacteria tend to protect themselves from the environment, harsh environment. So for that purpose, bacteria build a protective layer around themselves, which we call biofilms. And biofilms help bacteria to adapt to better survival mechanisms under stress conditions. So there are basically 5 stages that progress towards a stable biofilm. It starts with the attachment to the surface, then the formation of monolayer and production of a matrix. Then a colony is built, that is basically multi-layered bacterial cells, colony of multi-layered bacterial cells. Then it develops into a mature biofilm with different shapes such as mushroom and column and canopy shape, and detachment and reversion of tectonic growth starting a new cycle. And then these biofilms are detached and then they start a new cycle. So on Earth, um, when the gravity conditions are normal, the biofilm tend to produce a mushroom form biofilm, a mushroom-shaped biofilm. But in space condition or microgravity, the biofilm tend to be of column and canopy shape. Okay, so let's discuss some examples of instances of microbiologically influenced corrosion and low Earth orbit. So basically the idea is that how the biofilms are impacting and degrading the metal surfaces or metal equipments that are found low Earth orbit, like the International Space Station and different various equipments in International Space Station, such as water potable system. It is a metal equipment, and mainly the bacteria tend to produce biofilm around it that can degrade the water qualities, affecting the health of the astronauts. So, some of bacteria that were most frequently isolated from the potable water system on International Space Station were Pseudomonas, Methylobacterium species, Enterobacteriaceae, Bacillus, Propionibacterium, Corynebacterium, Staphylococcus. All of these were isolated from the potable water system on International Space Station. It was observed that biofilm was causing issue in water processor assembly of International Space Station. Changes in water pressure was detected, as you can see, between waste tank and water processor, mostly liquid separator, in 2010. It dates back to 2010, and yet we haven't identified a solution to it. It has shown that MLS, that is mostly liquid separator, inlet solenoid valve, was clogged by fungi and bacterial biofilm. Now you can see the importance of the topic. Space-based plant growth systems are available that can be degraded with it. The system has components. The disruption of these due to biofilms will lead to contamination of food, water, corrosion of electrical circuits and tubing, and mainly the health of astronauts, affecting the health of astronauts. So if you can see in this figure, the image shows the formation of biofilm inside the solenoid valve at the inlet to the water processor, mostly liquid. It was causing, it was blocked by bacterial and fungal biofilms, and it was causing changes in the water pressure in the water portable system. You can see these are clogged. Okay, this one is just an open channel, and all other are clogged by the biofilm. So the main species that was tending to produce these biofilms and corrosion, microbiologically influenced corrosion, was Bacillus subtilis. It is a gram-positive, motile, non-pathogenic, facultative, anaerobic microorganism. It produces biofilm under stress condition when a change in nutrient availability is observed. Bacillus subtilis is used as a reference organism for biofilms. So these are basically small motile roots. After the formation of biofilm, it forms long chains of non-motile cells that attach to one another and to the surface of the equipment by segregation of extracellular polysaccharide matrix that hold the biofilms together. So as we know that it is a representative, as we already discussed, that it is a representative genus of MICT search, the bacteria which is in genus Bacillus generates acid, which lead to the formation of corrosive compounds. The Bacillus produces corrosive metabolites such as ammonia enzymes and compounds that stimulate the production of corrosive biofilms. So the main aim and objective of the study. The aim of the study was to understand biofilm formation in spaceflight conditions under microgravity. So three main objectives were a part of this study. The first one was to identify the genes associated with biofilm formation in Bacillus subtilis, subspecies subtilis strain 168. To identify and map biofilm associated orthologs from other bacterial species. This was to get a collection of biofilm associated genes from all other bacteria. So ultimately we can get to a set of genes that are involved in this, and then we can produce a solution. Or at least identify it or present it to the industry. To analyze the fluctuations in gene expression under microgravity using NASA Gene Lab dataset, and to look how the expression of biofilm associated genes changes in space. Okay, so methodology. Our data sources included gene ontology database, UniProt, SubtiWiki, that is a reference database of Bacillus subtilis, and NASA Gene Lab data system. So we took a data set, identified a data set, RNA sequence data set, GLD as 185, Gene Lab data set 185, and then extracted the first few files and performed RNA sequencing of that data set. So first step was identification of model bacterial strain, that is Bacillus subtilis. Selection of the data set that I already discussed, that it was GLDS 185. Identification of genes associated with biofilm formation, extraction of these biofilm genes of all bacteria, now faster sequencing by ID mapping to get a collection of biofilm genes, and then BLAST to get the similar genes in other species. Gene expression analysis was performed using RNA sequencing technique, pre-processing, normalization, alignment, and differential expression. This led to give us some good genes that were involved in biofilm formation. Comparative analysis with some of the studies that were performed on the same GLDS and the same species. Validating the presence of novel biofilm, we identified some novel biofilm-associated genes in Bacillus subtilis transcriptome, and then enrichment analysis was performed to get to know the functional profile of the species. So fast forward, moving to the result section, identification of biofilm-associated genes. We identified 8 biofilm genes and their locus tags. These genes were performing. These are the genes that were performing or involved in the biofilm formation. So a total of 8 genes were identified that were involved in biofilm formation, and then a comparative analysis was also performed with Morrison et al. That used the same data set for identification of biofilm genes. And then we compared our genes with their genes, and we found that 2 of our genes were similar. Okay, this is my gene expression analysis GLDS 185 result. So this is basically volcano plot with a threshold of p-value, statistically significant genes with a threshold of 0.05 and log 2-fold change of greater than 0.5. So these are basically statistical tests that were applied on the data set during analysis. A total of 1,000 genes were differentially expressed, out of which 472 genes were underexpressed, while the remaining 582 genes were overexpressed. These are basically, the red ones that you see here are basically the significant genes. These are upregulated, while the left ones are downregulated, and the center ones are non-significant genes. Fast forward.
Hi, everybody. So I had to stop the recording. We have 10 minutes. These are 10-minute talks, and this recording seems to be about 20 minutes. So unfortunately, I do have to stop it short so we can continue on with our next speaker. So our next speaker for today is Frank Sobozensky. I hope I pronounced that correctly, from the University of York and King's College, London. So Frank, if you wouldn't mind turning on your audio and video and sharing your presentation.
All right. Hello. Thank you. Sharing my screen. All right. Can you see my screen and can you hear me?
We can hear you, and we do see your screen, yes.
All right. Wait a second. Just drag this one out there. All right. Still some overlap, but never mind. Hello, everybody. Thank you for having me. Today, I'm talking about explainable vision transformers on empowering MPPS microscopy data classification. This is work done in the AI/ML analysis working group. And first, I'd like to talk a little bit about the data that we are using for this. So this is based on the BPS microscopy benchmark set that is available on AWS to download. So if you go to that link, use that connection string there, then you can download the data set. If you do that, this is how it's basically will look like. So you get a meta CSV file within it, and that gives you an index of the images that are there, gives you some amount of dosage intensity values here, and a couple, and then the classification values as well. And this data set is fluorescence microscopy image data on individual nuclei from mouse fibroblast cells, and they have been irradiated either with high energy ions or X-rays. So these are the two classes, the particle types that you see here in the index file. This connection string that you see down there worked for me quite well if you're using a Mac to get the data. Now, if you download this data, you get an image folder with the index file, but basically just a folder of all the images. You can then use it and modify it for your machine learning, depending on what machine learning algorithm you're using. you may need to do some more tweaking to the data set. That's why we also uploaded this to Hugging Face. So Hugging Face is a repository that has become quite widely known as being the core center point for all things large language models or transformers. So every large language model is a transformer under the hood. And as you can see here, there's like, if you click on models, over 600,000 language models, all the latest ones that have been trained on millions of dollars from all the biggest corporations, they are there. And also on the left-hand side, you see all the categories that you can use. You can click on them, you can download them, you can fine-tune them, and you can further use them for your specific use cases. There's also a dataset option up on the top. So if you do click there and enter GeneLab, you will find the same dataset is on Hugging Face, and you can utilize this by copy pasting that dataset string on top that you see there, and just importing it via one line into your Python code. So for example, you just do a pip install of that dataset library, and then this is all you really need. And if you wish to use a different dataset, you can just change the name here. That's how easy it is to basically load the dataset in memory, and then you can split it into the different train test splits and so forth on. Having a look at the data exactly. So this is like how it looks like. There's then different images in the two different classes, X-rays and ions. They're a bit mixed up here. So here's a more clear view on the two different examples here. This has been used for a couple of machine learning methods to try to classify them. So, to my knowledge, all the traditional machine learning methods that have been used so far have just yielded unsatisfactory results. For example, I think the latest CNN just resulted in roughly around 50% accuracy, which is basically just random chance. So with me working heavily with transformers or large language models over the last few years, I really wanted to see how these new models perform on a simple classification on this one without even manipulating the images further. So there are no annotations inside the images that kind of highlight some features. It's just an overall classification of what is in the image, where it came from. Transformers have become quite prominent across domains, and there's, as you saw, 630,000 models and counting out there. All with specific features, but if you take them all away, under the hood is basically just three elements that make them work so well: positional encoding, attention, and self-attention. So positional encoding is just, if you take a language example, you make the model aware of what the order of the sentence is. It's different meanings depending on how the words are ordered in a sentence, for example. Attention is where the model listens in the context. If you think of a conversation and the model listens into the most important words for that particular thing it should look for, it will identify those and rank them and map them, for example, different languages, English to German. And self-attention is with the model learning the context of a particular word in the same sequence from the other words that are present. So if you think of a quick brown fox jumps over the fence, it learns that fox means it's an animal because of the context of the words. But if you think of a Fox News reporter quit his job today, you know it's not an animal, it's a person because of the context of the words. So become quite prominent in lots of fields, also going into self-driving cars and segmenting and so forth, millions or hundreds of thousands of different versions out there. The one we particularly were interested in using vision transformers. So these use similar architectures. They just, for the input here, input focus, they chop the image up and they feed the image as a sentence of images basically into the model. And then you can already see here positional encoding is applied and the other mechanisms can come into play as well. So we fine-tuned a vision transformer and we published this one. This is also available on Hugging Face now. And for that, I can switch quickly to my desktop. I hope you can still see that. And this is the website on Hugging Face. And there's some example images here that you can download. And you can drag it into the inference API. And it will load the model and perform an inference live here in that little API interface. But you can also utilize the code on Colab. So here you can see the results. There's an overall result of this model, which is 81%, which isn't great for medical the medical domain, but it's a good start compared to previous, and there's lots more that can be done here. This model is fine-tuned on the base patch model, so this is quite dated. So simply just changing the base model will already result in a significant increase, I believe. So here we have a code segment that just downloads example images, and all you need here is just to run this code in the Colab, and it will do the same thing. It will run the inference for you, which I've done here basically. So it downloads the images in the second code segment in respect of time. I'm not running this now, but you can see it gives you the same output. Just copy pasted this in here. We ought to also utilize weights and biases for monitoring your model training. So you can really train multiple models and then go back and pick the one that you are most fond of and take this one forward without risking picking the wrong one and having to go back and search for another model. Similar, so all you need to do for this model is just specifying here that string GeneLab MPPT. That's all you need for loading that transformer, and you can again fine-tune it on another task or running it over your data. I've mentioned weights and biases already. So concluding, the next steps that we want to focus on is now going to those mechanisms that transformers have and drawing out the attention maps where the model is actually looking for what it's deciding on in the features of the images, and you can do that by utilizing those attention maps. And they look like, if you can think of a heat map, and here we have a few examples from a different vision transformer that I was working on with the Solar Dynamics Observatory, and the middle image shows like a very early example of an attention map drawn out over the image inference. And on the right, you see the individual attention maps on the individual chopped up images when it feeds them into the model as a composite here. There's lots more to be done. So this goes into perhaps looking into changing the base model, as mentioned earlier, chaining the model perhaps up into individual tasks. So one transformer is looking at a particular feature and the other transformer takes that input from the previous one and performs a specialized task similar to modern RAG models that are out there. And of course, also model composition. So thank you very much.
Thank you, Frank. Our next speaker is Jian Gong from the University of Wyoming. So Jian, if you are on, if you can please turn on your camera and your audio and feel free to share.
Yes, I have a little problem. Hold on. Okay, I'm good. Thank you. Where it's going to a different screen, which is weird. Okay, sorry about that. So my name is Jian. I'm here representing the AI/ML AWG group on behalf of James Casledo and Lauren Sanders. So my topic today is tackle the incomplete data challenge in space biology by employing some of the AI/ML tools and also an idea called digital twin. So space biology confronts a critical issue at the moment is that we are challenged by limited and incomplete data sets during just because the space missions are complex and very expensive. So for example, in one of the research data set we are looking at, so this is a rodent research data set. We're looking at the mouth, especially look at the gene expression of the eye. And in this data set, you can see that, so these are different organisms. For example, this is one mouth, for example, and this is a sample from the mouth on the left. And then you can see we have analysis that is really scattered depending on the condition. Sometimes we have, in ideal condition, we want all of the data, and that can give us information tying between different causalities, right? But the difficulty is that we don't always have that. And so if you look at data set we're dealing with, you have, in addition to sequence data, we have images, we also have microCT. So these are additional modes of data sets, making it also a little more challenging. So I'm coming from a practical point of view and thinking, how do we approach solving this problem? And this has been what we've been thinking about for the past few months. So the research goal for the R9 project is really to evaluate how space flight affects the mouse retina, which is a model for human retina. And the hypothesis, the central hypothesis there is that fluid shifts caused by either microgravity or hypergravity represent a stress factor. And so this could induce neural anatomical changes to the eye, as well as the cell deteriorations in the retina through oxidative stress. So the idea is that if you collect gene expression data along with physiology data and looking at the image data of the retina, we can construct kind of find the reason what is happening during space flight. And so we need to compare a flight versus a ground control. So for this is a gene expression. It's rather complex graph of 4 different data sets visualized from the GeneLab visualization app on the portal. And so I just want to point out the complexity of data set in itself. We have different studies spanning over many years in between, like 2017, 2014, and there's also ground-only studies. And we're looking at, on the left, we are looking at the genes, individual genes that is highly expressed compared with the ground control. So we're comparing the differences between different groups. And the bottom labels are the samples we're collecting. And then you can see in this kind of heat map, you can see the differences between the ground and the space flight. The challenge here is that if you look, trying to compare this set of genes to this set of genes, they're rather different. In fact, compared to this, is also quite different. It's kind of hard to think about how can we use all of this data all together. So for example, I found one common gene that is highly expressed, but also is different between the between space flight and ground control, and this is a heat shock gene, a protein. So in the end, we are challenged with our ability to interpret this kind of data set and thinking through how to do this. There are really 2 objectives, in my opinion. Number 1 is we want to understand the cause. What is the reason behind the change in space flight? The other is rather important, is we want to develop and test remedies. For example, in a study in 2014, we found that antioxidants have some nutritional benefits that countermeasure the stress experienced in space flight, and so this is like doing a green tea kind of mix. But we need more of this kind of studies. But every one of them is a new condition, is a new set of experiment to do, new data to collect. So it's just becoming very difficult to do in a space kind of scenario. And so to answer this question, we really have to think through, like, how can we accelerate the progress in these kind of studies? And so the challenge is really not sufficient data. So can we synthetically generate data? Another one is, how do you integrate new data with existing data? So how do you make use of all the information we have? So I want to briefly talk about two areas of research. This is really a work in progress, so we are still working on this. So one is from the machine learning side, we are investigating GANs and VAEs. I will speak about exactly what they are, and then the digital twin. So GAN is really a data augmentation technique that is proven to work in especially for RNA expression kind of data sets. So that, for example, this study started with 50 RNA sequence samples, but they were able to generate 1,000 augmented samples from that 50. And from that, because we did that, the accuracy of the machine learning models has improved quite a bit. So going from 94, 70 to 98, 94. So there are real benefits doing this. So GNN really, in a way, it kind of fake the data starting from random input. But there are two machine learning models in there. One is called generator. Is actually going from the random input to generate a sample that later will be compared with the real data. And the other one is called the discriminator, which is really trying to compare the two to see what differences they are. And so by training two models simultaneously, we're trying to maximize the output until that we cannot distinguish the fake and the real anymore. And that really mimics the most of the statistical properties of the data set. The other idea is called conditional variation autoencoder, which is really also two CNNs. One is the encoder, the other decoder. They work together to trying to find features in the data in terms of the distributions, the mean and the variance, and trying to sample from here and regenerate. And here we have a lever, which is the condition. We can use that to tune the data so that we can produce 30 different kind of data sets depending on the condition. And this condition can be thought of as something that is real, almost like a real thing. So this kind of work also works and has demonstrated to be working in gene expression data sets. I'm going to jump because of the time about digital twin and why we want to do digital twin. So in the definition, it's really a dynamic and self-evolving model or simulation of a real subject. It can be a part, can be a machine, can be a process, or can be a human or parts of a human. So it's really flexible how we define it, but the real power of having this is that we can start asking what if scenario questions and potentially saving cost in the long run for space missions. And we don't have to launch a new mission. We can investigate from the data we have, make the most use of the data. So I want to thank the Digital Twin subgroup, especially James Casledo and Lauren Sanders, JJ Hastings. And if you're interested in the project, send me an email. We can hook you up and we can work together. Thank you.
Thank you so much, Jian. That was great. And our next speaker is Nilufar Ali from Boise State University. So Nilufar, if you are on, may I please ask you to turn on your audio and video?
Yeah, I just did. Can you see me?
We sure can.
It's going to the different screen. Do you see the presentation? Sorry.
Not yet. Not now.
No.
Yes, but they're not in slideshow mode. Now they are. Perfect.
Awesome. Welcome, everyone. Today I'll talk about how space travel can trigger Parkinsonism-like molecular changes. So there are observable macrostructural changes in the brain circuitry and microstructural changes along the somatosensory, visual, and other regions of the brain after space flight. However, it is reported that there is a net change in the left caudate nucleus and the basal ganglia after spaceflight. And this region is also important for Parkinson's disease. And as you know, that Parkinson's disease is also a motor neuron, and also a motor disorder where there is the postural instability, tremor, rigidity, and slowness of movement is what the patients suffer from. It originates from the degeneration of neurons. If you can see my pointer in the substantia nigra region of the brain, and the output of these neurons are in the striatum. So due to the loss of this nigrostriatal pathway, there's a loss of dopamine neurotransmitter in the brain, which disrupt the balance between the direct and indirect pathway in the basal ganglia, the same region which is also reported to be affected post spaceflight. And this disruption leads to the motor coordination impairment. So the etiology of this disease is not very well known, but the key hallmarks for this disease is mitochondrial dysfunction and oxidative stress, which is also one of the hallmark for the space flight. So what are the evidences, or how do I come to this, this idea of molecular signatures similar to spaceflight, which are also observed in PD? So this paper by Willian and group, which is a multiomics analysis from several astronauts data post spaceflight, which gave it's like the landmark paper, which tells us that mitochondrial dysfunction and oxidative phosphorylation are the key hub for all the genes which are going upregulated or downregulated post spaceflight. And the GC analysis, or the gene set enrichment analysis of all these genes from the in vitro cultures in space has shown that the OXPHOS related genes are also common in the neurodegenerative diseases. And when you look deeper into this genes, you see that there are Huntington genes, Alzheimer's, and Parkinson's disease. Like so many of these genes, the 142 PD genes are also affected post spaceflight, which are common in spaceflight and also in PD. So as my interest lies in the PD, I looked into more details of what are these genes. So mostly these are the mitochondrial oxidative phosphorylation genes. However, if you look at the yellow dots, these are some of the cardinal PD related genes, and some of them are familial PD genes, which are also differentially regulated after spaceflight. And what do these genes do? Like, what's the molecular function? So these genes are mostly, as you may expect, are oxidation reduction pathway related genes. And some of them, if you look at the next slide, it is the most of them are related with mitochondrial electron transport chain complexes. Some of them are endopeptidase involved during apoptosis, and the ubiquitin proteasomal system related genes. So if you ask me that, are there any other parallels other than the mitochondrial dysfunction? So this is like a chart where you can see the green is spaceflight, and these are the conditions which are common seen between spaceflight, Parkinsonism, the lighter blue shade is Parkinson's disease. And these are Parkinsonism, which is also related to Parkinson's disease, but they have some added clinical signatures. And these are the other neurodegenerative diseases, including Alzheimer's, Huntington, ALS, and prion. So all of these genes, all of these multifactors or the conditions have overlapping, they overlap with each other. However, if you look at the yellow field boxes are a yes answer, and the white field boxes are no answer. The ones with questions are the one that we still don't know about, like are there aggregated protein formation in the brain or elsewhere after spaceflight. So if you look at this chart, you see that spaceflight and PD have more commonalities than any other signatures. So Interestingly, other researchers have already shown that in C. elegans, for example, dopamine level goes down under microgravity condition. And in mice, it is also shown that the tyrosine hydroxylase, the gene which metabolizes dopamine, that also goes down in substantia nigra region of mouse brain. So these are some of the key clues to look into the PD pathways and what's going on in the PD affected region in under microgravity or in spaceflight. So we try to do experimental validation of this finding, and we use simulated microgravity clinostat, and we grew SHS5 neuronal cells. We matured them into postmitotic dopaminergic neuron, differentiated them, and we exposed them under simulated microgravity for 72 hours. Interestingly, we found that under simulated microgravity condition, after 72 hours, there was a reduced population of the cells. And when I tried to look at the expression level of all those the genes which we found from gene lab, TH level as absorbed by the others, we found that the expression of TH level goes down drastically, and the other genes like LRRK2 parkin were also affected. And this is very preliminary data. We are also validating it with other genes and increasing more samples. Next, we tried to look at mitochondrial therapeutics. We tried to work with, we started with MitoTempo, which was available very easily. And we saw that a pre-treatment with MitoTempo before simulated gravity exposure kind of ameliorated the effects. So we looked at the population of cells. There were increase in the cell population as observed. The red is the simulated microgravity and green is the simulated microgravity, but treated with MitoTempo before. So you see that the population was recovered. And if you look at the neurites look more beaded and swollen, which is like a typical hallmark of oxidative stress in cultured neuron and under simulated microgravity condition. However, in MitoTempo treated group, we see that the beading was gone and the neurons look more healthier. Next, we looked at the mitochondrial health related assays, such as the mitochondrial membrane potential. We found that under simulated microgravity, the mitochondrial membrane potential goes down or decreased. However, with the treatment group restored the mitochondrial membrane potential. Mitochondrial ROS goes up under simulated microgravity, and the ROS is kind of ameliorated or goes to the normalcy after MitoTempo treatment. So why do we care? Like, why do we want to study this? So all these results which we are seeing from today's talk and from my previous pointers, that all these are coming from the short-term space flight and not beyond the lower orbit. So we may assume that as we move on to deep space mission or the long-term space flight, these changes, these subtle changes which are all reversed back to normalcy in most conditions can be permanent, and they can affect the mission critical goal and health of the spacefarers. So there is more opportunity in this, like in this field, where we can do more research related to this long-term spaceflight stressors and the dopaminergic neuronal loss, specifically focusing on the regions affected in PD. And is there any behavioral change or motor change that we observe when we keep this mice for, if we are studying it in mice, if it goes and shows those kind of problem in behavior and motor changes. So disease modeling could be a good opportunity where space-flown mice can be used to model Parkinsonism, and also a drug testing platform. The space-flown mice and in vitro cultures could be also used for testing the therapeutic as well as mitochondria-targeted interventions. So in conclusion, the in vitro validation of, we did in vitro validation of this multiomics, Parkinson's disease and spaceflight mitochondrial hypothesis. And we saw that at least with one drug in the in vitro culture, we saw this, the effects were reversible, although these were not exposed to a long-term spaceflight stressor like the GCR, the cosmic radiation that we would imagine. And the Gene Lab, Multiomics AWG, we have formed a PD subgroup where we are studying molecular markers common to the brain-related OSDs and PD markers that are obtained from Michael J. Fox Foundation PPMI initiative database. New collaborators are always welcome. You can write to me if you are interested in joining the subgroup or even want to collaborate. I would like to acknowledge Dr. Afshin Beheshti and the team of the NASA Gene Lab Multiomics PD subgroup, Dr. Graham Pikian and Lab, Max Mortimer, Devin, Catherine, and Dr. Brad Morrison. I'd like to take the questions in the end. Thank you.
Perfect. Thank you, Nilüfer. Perfect timing. And we have our final speaker of this session and of the event for today, and that is Jet Ritz from the International Space University and Karolinska Institute. So Jet, if you are on, if you wouldn't mind turning on your audio and video and sharing.
Yes. Okay. Can you see my slides?
We sure can.
Okay, perfect. So then welcome to my presentation about a transcriptomic approach to understand pharmacogenetics of mice exposed to space-derived conditions. In this presentation, I'm going to present a part of my master thesis that I'm currently doing at the International Space University. And before I talk a bit more about my project, I would like to give a bit of a background so that we're all at the same page. So as you heard before, there are these 5 hazards of human spaceflight that were defined by the NASA, like space radiation or isolation and gravity fields, or more like the absence of it. And these hazards cause physiological adaptations and consequently problems like sleep deficit or space motion sickness. And these and other issues lead the astronauts, just like most people, to take medications. And when taking medications, there are always multiple things to consider. And one of these things is the effect of the body on the drugs, which is called pharmacokinetics. And this includes multiple phases, like the absorption, where the medication enters the systemic circulation, the distribution throughout the body, the metabolism, where the medication is transformed and made more water soluble to allow for the last phase, which is the excretion, so the elimination of the medication from the body. In this presentation, I'm going to concentrate on the metabolism, since my work is mainly focused on that part. And so when and where does it happen? So when the drug is given orally, it dissolves in the GI tract. It's given orally, then it dissolves in the GI tract. And there already some enzymes begin the process of metabolism. And then when it's absorbed into the portal vein, it enters afterwards the liver, where the majority of the metabolism happens. And most parts of the drug get inactivated here, and just a fraction will arrive finally in the systemic circulation. And the metabolism in the liver is split into 2 phases. The first one, which is the functionalization. So you have either, for example, reduction or oxidation reactions. And in phase 2, it's the conjugation, because there are hydrophilic groups added to the drug molecule to further increase the polarity. And both phases are defined by enzymes that catalyze the reactions, and they influence the individual response to drugs. And here just some examples. For example, the CYP gene family, or here DUGT. And because they're so defining for the metabolism, and with it, for example, for the efficacy of drugs, I studied their expression in space versus on Earth. And so my research question in my project was how do space-specific environmental factors influence the expression of pharmacogenetic relevant gene families, with a focus on drug metabolism. And with it, I aim to I aim to identify critical medication carried to space and also identify key genes with significant alterations in their expression to finally optimize medical interventions during space explorations. And to achieve this goal, I downloaded RNA sequencing data from the NASA GeneLab database, and I only used data sets specifically for the liver and where mice were sent to space. With this data, I did two main analysis steps. First, the differential gene expression analysis, where I looked at genes that are differently expressed between space and Earth. And then I filtered for genes that are interacting with drugs with the help of a table from the FDA and the drug gene interaction database. And further, I filtered for genes that are encoding enzymes of either phase 1 or phase 2 of the metabolism. And then in the second step, I did a gene set enrichment analysis where I looked at gene patterns instead of single genes. And these patterns or gene sets I created by using information from the DGI database again. And in this case, one gene set included all genes interacting with one drug. So I want to show you some results. So here you see a table with the drugs on the right side and the number of differentially expressed genes interacting with them on the left side. So for example, for this drug, you have 52 differentially expressed genes that are interacting with this drug. And as you may see, the first ones are mostly drugs used for cancer treatment. But since they're not relevant for my project, I specifically looked at drugs that are carried in space, and therefore I did some literature research and I marked them in red here on this table. So you can see that acetaminophen is the drug with the most genes interacting with it that's also carried on in space. And then coming from the drugs, let's move to the gene side. So these tables describe how many drugs interact with the differentially expressed genes named on the left side. And you can see that mostly CYP genes are present, and I also filtered here specifically for genes that are in the metabolism and have them here marked in red. And as I said before, you see that it's mostly CYP genes, which was expected because they already on Earth have high influence and they're metabolizing a lot of different drugs. And a final last table. So looking deeper into metabolism, I created this table which shows for phase 1 and phase 2 the differential expressed genes that encode enzymes of that phase. So you can see that here in this first row and here. And we see one, and then I matched drugs that are connected with these enzymes, so they're metabolized by these enzymes with them. And here I only show interactions with drugs that are carried on in space. And we see one name again very often, which is acetaminophen, and it interacts nearly with all differentially expressed genes that encode enzymes of either phase 1 or phase 2. And overall, we see that we have a higher variety in the second phase of genes or enzymes, but we have more drugs that are connected to the phase 1. So we have a higher influence in phase 1. And what can I conclude so far from my project? So first of all, I saw that acetaminophen is the drug that has the highest amount of differentially expressed genes from the drugs that are carried in space. So it has the most genes that are connected with it that are different between space and Earth. I saw when you look at the genes that it's mostly CYP genes which are altered through spaceflight and they have a very high influence. Then I saw that most drugs are influenced in phase 1 drug metabolism even though the variety of genes is higher in phase 2. These are so far my evaluations, but the analysis is still ongoing. And finally, I want to thank for your attention, and I would like to thank my supervisors for their supervision and guidance throughout my final master semesters and master thesis. And since I'm close to finishing my master thesis, I'm currently looking for an opportunity to continue my exploration afterwards in this field. And if any questions popped up during this presentation, I'm happy to answer them now or per email later. Like, you have some emails.
Thank you so much, Jet. That was a great presentation. Excuse me a second. If I can please ask all the speakers in this category to turn on your cameras. There are quite a few questions. All right. So Alex, we'll start with you first. What single cell data did you use to pre-train the autoencoder?
Yeah, sorry. I should have explained that during my presentation, but I'm sharing the paper that describes the single cell data that I used for pre-training the autoencoder on the chat. But this is a framework for all other GeneLab data sets. So in our case, we're using plant, we're doing analysis on plant roots. So we use this high throughput single cell data set for plant root. But if you're using different organisms like mice or something else, then you can use a single cell data set that you want to use on the to pre-train this sparse autoencoder. But yeah, thank you for the question.
All right, we have one more question for you. How did the results from the sparse autoencoder feed into your downstream analyses and biological findings?
Good question. So our main contribution is to provide this method or open source framework for everyone to use for sparse autoencoder. For us, we retrieve the data representation from the trained or fine-tuned sparse autoencoder and put it into use like ensemble clustering. To for us, the downstream task would be the clustering, but it could be used for other downstream tasks, for example, like prediction or else. And I also explained about post-pipeline analysis, which is our suggested post-pipeline analysis using this using the clustering result that you got or from the data representation. Which from the post-pipeline analysis you can connect to your further research, such as like wet lab experiment or else. But it's your choice or the researcher choice to use the data representation or clustering result that you get from the framework. Thank you.
Thank you so much, Alex. So our next question is for Frank. Frank, are you on?
Yep.
Okay, perfect. So Frank, why do you think the transformer performs better than traditional classifiers on the microscopy task?
Yeah, thank you. That's a great question. And I should have probably explained this in more detail. Apologies for that. So in general, you wouldn't need to throw a state-of-the-art vision transformer model at an image classification task. So if you have like a simple, straightforward images, cats and dogs, or something not complex in your image, traditional models may work very well on that. However, this is a particular kind of data, and we have used traditional models, as I perhaps highlighted in the talk. CNNs, for example, have been used, and they have unsatisfactory results so far on the accuracy ranging around 50% there. And transformers themselves have shown that they are up there in the best ranking models for some of those tasks. And the features that they bring may make it robust that more traditional models may suffer from. And by simply not doing any adjustments to this, we already resulted in 81%. Another reason why I think they I chose those is simply the attention mechanism, because you can draw it out. You can look at the model, what it is doing, why it is deciding on that one. It may not decide on what we wanted it to decide on. It may pick something up that we are not aware of, and it will highlight that why it is deciding on this particular feature. So, but we didn't do anything. So it was simply just a plain transformer trained on a very dated base model, actually. And that already kind of got us to that percentage in terms of accuracy. And we're taking it now to the next stage and investigating it and tweaking it and this sort of stuff.
Awesome. We have a couple more questions for you here. The next one is, are there any adjustments that you think would help the transformer perform better? Something closer to medical grade classifiers?
Yeah, yes. So going back to the base model, again, this is like a 2-year-old, one of the very first vision transformers that came, were published, that we used for fine-tuning this one on. So simply just swapping that one out will bring additional features and additional performance, I believe. And then there's lots of parameters and lots of things you can do. So the toolbox is almost endless. So it comes close to also Alex's work. So you could do late representations. You can can feed this into the model. You could see where the model is looking at once you look at the attention maps, and then you can direct it. So you can highlight certain things and say that don't look at this, or mask it out, basically. If you think like, oh, the model is always looking into something it shouldn't do, so you can direct it, don't look there, go into the other direction. So that's a couple of things to do there. And yeah, lots of parameters and hyperparameters to be trained in general, but we haven't done any of this. It's just simply a base model at the moment. Yeah.
Okay, and one more question for you. This is very exciting. What plans do you plan on, or do you have any plans on using these image classifying and interpretation tools for other levels of images, like biosystems, microbes, plants, animals?
Yeah, absolutely. I mean, the options again are large, and there's tons that can be done chaining those different models together. Once we improve those, we can, as I mentioned, like highlight them in different tasks. One is maybe using specialized detection methods on this particular data set, the other one on that one. That can be done. And also in terms of medical grade, there's an effort currently that kind of treats language models as reinforcement models. So you have a mechanism called direct preference optimization that leans on a human in the loop feedback input, basically, to the model as well. That's additional steps that can be taken in various areas. But in general, yes, but every particular use case obviously to look at the intricacies. So there may be certain things you always need. You cannot just throw it in general at it. So if you need to have a look at plants data sets, maybe different from the other ones, of course, and we need to adjust and tweak and this sort of stuff. But the beauty of transformers is you can fine tune them in different steps and can build them up on different aspects.
Great, thank you. So the next couple of questions are directed for Gian, if you wouldn't mind coming on camera.
Yes, I'm right here.
Okay, first question is, have you taken the difference in mice lineage into consideration when looking at the different results?
That's a great question. No, we haven't done that. Lineage. Yeah, I know there is some information regarding the species, but in my heat map, you see there is sort of a tree on top and below. Those are, I think, sort of like relationship maps, somewhat like a phylogenetic like tree kind of thing, connecting in between different things. So you can take that and think of it as a lineage in a way, but that's respect to that protein. But I would love to look into the mouse itself. Thank you for the feedback.
Another question we have here is, could this type of analysis or predictions also be done having only the complete genome, like DNA?
Currently, these are, so the reason we do RNA expression is really to compare spaceflight versus ground control, right? So that's like the thing we are targeting. So what is the functional changes in terms of RNA expression in space that is activated? So if you do the DNA, I think that would be more useful for like a longer term time study to see how maybe a different organism, I mean, like if they reproduce and keep going, and then we can track the DNA of the different generations. That would be really cool. I think that can be more useful for things like plants and maybe smaller animals that reproduce much faster. So we can do that kind of study. That's what I'm thinking. So yeah, thanks.
Great, thank you so much. Our next set of questions are for Nilufar.
Yeah, I'm here.
Okay, so first question for you is, are there any nutritional or dietary interventions that you think would be worth testing?
Yeah, I would say that for PD, it is known that exercise, kind of cardio and other high throughput exercise helps. And there is not like known dietary restriction or anyways, astronauts, they do get a specific diet. So I don't know like how much restriction can you add on to that, but supplements like antioxidant supplements, vitamin D and several other vitamin supplements helps. And as one of the speaker, I think Veronica presented, that exercise is also something that is time consuming. So anything that can help reduce, or generate exercise like feature, like low intensity vibration, for example, that could help presumably in this condition. So yeah.
Great. We have one more question for you here is, for future students, do you think that brain on a chip or brain organoids would be useful? Oh, sorry, for future studies, not
Yes.
students.
Yes, the answer to that is definitely. We are looking forward to do any kind of like experiments that, so for brain organoids or brain on chip, that is very easily translatable if you are performing experiments in the International Space Station. So yeah, in future, we plan to do these experiments on chips if possible. And if anybody has any, like if you want to collaborate with us, feel free to reach out to us.
Wonderful, thank you. And we have a question for Jet, if you wouldn't mind coming on camera.
Yeah.
So your question is, very nice work. Is it possible to develop more robust drugs that are not so strongly affected during phase 1?
Maybe, but I think until now we can't say enough, because like my research that I did is very at the start, and we need more knowledge on where the CYP genes or where enzymes of the phase 1 target exactly, to look what we can do about it. But currently you also need to look at splicing and which variation of the enzyme it is in the end. I think we don't know enough to think about that step yet.
Great, thank you so much. And Gian, we have one more question here, if you wouldn't mind coming back on. I missed this question, I apologize. Gian, are you still on? Okay, perhaps he left. So thank you all for your time. I'm going to turn it back to Lauren Sanders for some closing remarks for day 1 of our 2024 AWG Symposium.
Hello, everyone. Can you hear me?
Yes, we can.
All right. For some reason,
Okay, I think we lost Lauren.
sorry, I'm back. Zoom is not playing nicely with my NASA computer today. But as long as you can hear me, thank you so much, everyone, for an unbelievably fantastic first day of our symposium. It was great to hear all of the wonderful work that you've all been doing. And we're looking forward to a second day of talks. So please join us tomorrow. And thank you, Lavorka, for moderating. See everyone tomorrow.
Bye, everyone. Thank you.